So we will start to discuss process of bacterial transcription, which we can break down to three basic stages: initiation, elongation, and termination. During initiation step, transcription apparatus assembles on the promoter and begins RNA synthesis. The position of the start site of RNA synthesis is determined by the consensus sequence, which we will talk a little bit more about in next slide. Now remember, during transcription, RNA polymerase does not require primers, so no primers are needed. So once the initiation is completed, elongation takes place. During elongation, DNA is ready through RNA polymerase, which unwinds DNA and adds new nucleotide to the 3' end of growing RNA. So new RNA strand is synthesized in 5 to 3' direction, and it is anti-parallel to template DNA. So once a transcription unit encounters termination sequences, it does recognize the end of transcription unit and the RNA is separated from DNA template. In previous slide, I mentioned that a transcription apparatus recognizes consensus sequences in bacterial promoters. So what are consensus sequences? Upon examination of many E. coli promoters, it was shown that there are common common sequences that you can find in most of the E. coli promoters, and these so-called consensus sequences play a very important role in initiation of transcription. Now, when you look on these consensus sequences, basically you can see that in first position here you have T, consensus sequence timing. Sometimes you can have exception, however it's very rare. If majority of promoters have particular base at that position, so that nucleotide take place in consensus sequences. The same apply to second position. Sometimes you can encounter something which is labeled Y. Y refers to any pyrimidines at that position. Similarly, R indicates purines. Sometimes at certain position in promoter, because you go in consequent sequences, there is not particular bases added, and if there is no particular base, you label that position as N. And you can also encounter annotation such as this one, C/G, which means that at that particular position you can have either cytosine or guanine. Now, next slide we will start to discuss initiation, and you will see importance of these consensus sequences in bacterial promoter. So initiation of bacterial transcription comprises promoter recognition, formation of the transcription bubble, and creation of the first bond between ribonucleotides and escape of the transcription apparatus from the promoters. You remember in bacteria we have only one RNA polymerase, and basically that polymerase recognizes consensus sequences -10 TAT, which sometimes you can find under the name of Pribnov box, and -35 consensus sequences. So bacterial holoenzyme binds to these two consensus sequences, and importance of the sequences have been defined when mutation have been introduced which completely annihilated the transcription or completely inhibited transcription. So any mutation in these two consensus sequences would inhibit RNA transcription. And it is because holoenzyme binds to this consensus sequences. So upon binding and recognition of the consensus sequences, transcription apparatus start to unwind of the DNA, and this unwinding starts right here at -10 consensus sequence. And from there, it does extend downstream of these consensus sequences. Most bacterial promoters are located upstream of the coding sequence of the transcription start site. So what positions all transcription apparatus, that holoenzyme, to the transcription start site is, you remember, sigma factor, which completes the holoenzyme. So let's now summarize transcription initiation. As we discussed before, core RNA polymerase associates with sigma factor and form holoenzyme, which binds to consensus sequences within bacterial promoter. The orientation and spacing of consensus sequences on DNA strand will determine which strand will serve as a template. Upon binding those consensus sequences in the promoter, holoenzyme enables tight binding and proper positioning. Respectively, this proper positioning is directed by sigma factor. What does it mean, proper positioning? So holoenzyme is positioned that basically it starts transcription at that start site, which we label +1 site. So upon this proper positioning, which is enabled by sigma factor, a nucleoside triphosphate, which is complementary to the DNA at the start site, is added to the template, following by cleavage of two phosphate groups, which creates a RNA nucleotide, which will be the first nucleotide of this newly synthesized RNA, which will form the 5' ends of the RNA. This also triggers release of sigma factor, and releasing of sigma factor from holoenzyme enables polymerase to move beyond the promoter and initiate elongation. Very often during the initiation, RNA polymerase actually does not proceed completely. It just generates a short transcript from 2 to 6 nucleotide. And while it's still bound to the promoter, this process we call abortive initiation, and you can encounter abortive initiation in bacterial transcription but also in eukaryotic transcription. And often after several abortive attempts, polymerase start to synthesize an RNA from 9 to 12 molecule lengths, which allows the polymerase to move to the elongation stage. As we discussed before, at the end of initiation, RNA polymerase, that holoenzyme, undergoes conformational change. This conformational change triggers release of sigma subunit, which allows RNA polymerase to move downstream beyond the promoter. And as it moves downstream, it adds nucleotides approximately at the speed of 40 nucleotides per second if the reaction is kept at 37 degrees Celsius. Now, the transcription takes place within so-called transcription bubble, which is comprised of the short stretch of approximately 18 to 20 nucleotides. So, as the transcription apparatus moves, unwind DNA, synthesize RNA, you can imagine that it would generate positive supercoiling ahead of the transcription bubble and negative supercoiling behind it. So, therefore, topoisomerases are required to relieve the stress associated with this unwinding and rewinding of DNA. Last thing which I would like to mention to stress is that RNA polymerase is quite accurate in incorporating complementary nucleotide into the growing RNA strain. However, errors can happen. And similarly to DNA polymerase, RNA polymerase has capability of proofreading. Proofreading ensures that RNA sequence is complementary to DNA template, so it assures proper transcription of the DNA template. RNA polymerase is adding nucleotides to the 3' end of the growing RNA molecule until it transcribes termination sequence. So, transcription stops after the terminator has been transcribed. So, once the RNA polymerase stops synthesizing RNA, it has to release RNA from DNA template. In bacteria, we have two basic kind of terminations, so-called row-dependent and row-independent termination. Row-dependent termination depends on two things. There is a so-called row binding site on synthesized RNA. So row terminator binds to RUT sites and moves towards 3' ends of synthesized RNA. So whenever RNA polymerase encounters terminator sequence, it pauses. And once it pauses, row is catching up with the polymerase, and it uses its helicase activity and unwinds DNA from RNA, so it breaks down DNA RNA hybrids and releases RNA, newly synthesized RNA. Row independent termination depends on the inverted repeats, which are followed by approximately 6 adenine nucleotides right here. So the inverted repeats are transcribed into RNA and form hairpins. When hairpins is followed by a string of uracil, which is transcribed from those adenine sequences in DNA, this destabilizes DNA-RNA pairing, and RNA transcript separates from template, terminating thus transcription. So these are the two major mechanisms by which transcription is terminated in bacteria, row-dependent and row-independent. As the last thing I would like to mention here is that in bacteria, there are differences in how genes are transcribed from the eukaryotic transcription. Usually, a group of genes is transcribed into single RNA molecule, and we call this RNA molecule polycistronic. And it's a product of a single terminator which is present at the end of group of several genes which are transcribed together. You don't find usually polycistronic RNA in eukaryotics. It is characteristic of the prokaryotic gene transcription.